We'll now look at the mechanisms that lead to the formation of the neural tube, beginning with a process of primary neurulation. We'll also examine the consequences of defects in some of these mechanisms. Neurulation begins at more anterior regions of the embryo, and in these representations, which show the chick embryo, you can see that the anterior or the cephalic region of the neural tube is already undergoing neurulation, while gastrulation is still happening at the posterior region where we have the primitive streak still present at that location. So as you can see, the neural plate has already folded into a groove in the anterior region while the primitive streak is still present in the posterior. And I want to clarify that doesn't mean that the most anterior portion of the neural tube closes first. In the chick, the closure begins at the level of the midbrain, and then it zips up, the neural tube zips up from both directions. As we'll see later, neural tube closure in the mouse and in human begins at different closure points. One common characteristic is that the most anterior and the most posterior regions, as we'll see in a moment, are the last portions of the neural tube to close. The neural plate is induced to form by the notochord, which is that rod of mesoderm that's located under the ectoderm. It's also induced to form by the head mesoderm. Now, once the neural plate has formed, as it has in this figure, the first stage of primary neurulation involves the elongation of the neural plate along the anterior-posterior axis. This is not evident in these pictures, but what is meant by elongation is the lengthening of the body axis, which begins during gastrulation. And that process involves cell divisions that occur preferentially along the anterior-posterior axis. The neural plate then begins to fold. The folding of the neural plate starts at the midline with the formation of a hinge point that's known as the medial hinge point. And in order for that folding to happen, some of the cells need to change shape to form the hinges. As you can see, the cells at the medial hinge point have a wedge shape, and these cells are anchored to the notochord. That anchoring to the notochord is important to promote the formation of the neural groove. And it's also thought to be important for ensuring that the neural tube folds inward rather than outward. After the medial hinge point leads to the formation of the neural groove, the epidermal ectoderm is going to induce the formation of dorsolateral hinge points. These are also, as you can see here, wedge shape. These dorsolateral hinge points are anchored to the epidermal ectoderm, and the association to the epidermal ectoderm is thought to be another mechanism for preventing the neural tube from folding outward. The ectoderm also pushes toward the midline, and that brings the neural folds together. The cells of the medial hinge point and the dorsolateral hinge point acquire the shapes that allow them to form those hinge points as a result of three major reasons. There are changes to the actin cytoskeleton that lead to the formation of an apical constriction, which is what you're seeing here. So this is a constriction that results as a result of contraction of actinomyosin complexes that are located at the apical side of the cell. There's also the movement of the nucleus toward the basal side of the cell, so that allows the apical side of the cell to be more narrow than the basal side. Finally, the amount of proliferation that happens in the neural folds increases, and that increased proliferation is thought to promote the bending at this location. Eventually, the neural tube closes at the midline, so the neural folds associate with each other, and the epidermal ectoderm from one side associates with the epidermal ectoderm from the other side, so that covers the neural tube. As that happens, neural crest cells delaminate from the dorsal aspect of the neural tube. And I should mention that I use the word delaminate here, even though these cells of the neural crest are not separating as a sheet of cells, but rather as individual cells, because this is the term that is historically used to refer to the separation of the neural crest from this location. As you saw, in order for the neural tube to fold and to close, hinge points have to form both in the midline and by the neural folds, and the formation of these hinge points requires changes to cell shape. But what is it that allows these changes to happen, and what is it that allows them to happen where they do, at the precise locations where they need to happen? Well, it turns out that the levels of BMP and to a certain extent sonic hedgehog are important for regulating the formation of hinge points. The notochord is known to be needed for inducing the formation of the medial hinge point. It's also known that sonic hedgehog is secreted by the notochord and that it induces the formation of floor plate cells. And these are cells that include the cells that form the medial hinge point. And they're important for determining the types of cells that are going to form along the dorsal ventral axis of the neural tube, as we'll see later. So the notochord induces the formation of the medial hinge point and induces the floor plate cells. But in animals who are knockouts for sonic hedgehog, the medial hinge point actually still forms. So that suggests that there are other signals that come from the notochord that are important for promoting the changes in shape at the midline of the neural plate. And the current evidence suggests that the most important factor mediating the formation of both the medial and also the dorsolateral hinge points are the levels of BMP. The regions that surround the neural folds secrete the BMP inhibitor noggin, and noggin is also secreted temporarily, at least, by the notochord. And the presence of noggin at these locations inhibits BMP signaling, and this has been shown to be critical for the formation of proper hinge points. In fact, in mice that are noggin knockouts, as you can see here, the neural tube doesn't close properly. So that would suggest that BMP has to be inhibited in order to allow the neural tube to close. But it's actually not that simple. Because as it turns out, expressing a BMP receptor that cannot be activated, so a dominant negative receptor, leads to the formation of exaggerated hinge points and also ectopic hinge points, so hinge points that form at locations where they should not form. On the other hand, expressing a BMP receptor that is always active, even in the absence of BMP, prevents the formation of hinge points. So intermediate amounts of BMP signaling in the neural plate are required to ensure that hinge points form and that they form only at the correct locations and that they have the appropriate shape. And these results are represented in this figure as well. So how is it that intermediate levels of BMP lead to the formation of the hinge points? Well, higher levels of BMP lead to the recruitments of proteins that stabilize the junctions or the connections between cells. And that prevents folding by keeping the size of the cell even across the apical and the basal regions of the cell. At lower levels of BMP, such as in the presence of noggin, which is represented here, the cell junctions are destabilized, and that allows the formation of an apical constriction by actin and myosin filament. So that allows the cell to change shape. This figure summarizes how different morphogens regulate the formation of hinge points. The epidermal ectoderm expresses and secretes BMP, which is shown here in green. But this BMP is inhibited by noggin, which is expressed by the dorsal neural folds. Now, since BMP inhibits the formation of the dorsolateral hinge points, the inhibition of BMP by noggin, that's secreted by the neural folds, allows these hinge points to form properly. The notochord, which is located under the neural tube, or the developing neural tube, secretes sonic hedgehog, and that establishes a new sonic hedgehog secreting center or signaling center at the floor plate of the neural tube, so the ventral side of the neural tube. Now, sonic hedgehog inhibits noggin, as you can see here, and in doing so allows for just the right amount of noggin to inhibit BMP. This is so that you have just the right amount of BMP in order to prevent the formation of ectopic or exaggerated hinge points.